Hi, I'm Jay Overholser. Let's see. And I'm going to be speaking more about our xMAP work that we did with this project. As Linlin spoke, we start out using an ELISA assay, and in the data he showed you what would be kind of the equivalent of the reverse, where he had the PD-L1 in the liquid phase and PD-1 on the plates. And he had found out that the reverse did not work as well, putting the PD-L1 on the plates. So first thing we did was we made beads. So the first couple times we did this, we used the xMAP antibody coupling kit. And this is just a quick look at the workflow on that. So it comes as a very nice kit from Luminex. And you're able to take beads, and these are Magplex beads. They have different code numbers on them, of course, that correlate with the machines. And then you basically isolate the beads with a magnet so you can do washes. Very nice. We activate the beads using sulfo NSS and EDC and add that to the solution of the tube. The tubes are reacted for 20 minutes in the dark on a rotator. Again, we isolate and wash them. You can add your dissolved protein, in this case, either the PD-1 or the PD-L1, to the beads. Incubate for 2 hours in the dark on the rotator again. And then we isolate the beads with a magnet, wash, resuspend the beads in the wash buffer. There's a small amount of BSA in the wash buffer, so that also will block any reactivity and keep things from continuing to stick to the beads. And then you dilute the beads after counting them, and we diluted them to 2 million beads per mil. Again, that workflow was using the kit. Instructions, including the buffers and things like that, can also be found in the xMAP cookbook, which some of you may be familiar with. And this actually starts on page 18, and it's called the Carbamide Coupling Protocol for Antibodies and Proteins. With this study, we use the Luminex xMAP and Teleflex. Okay, so this is showing a direct assay. Again, so with this one, this would be taking PD-L1 beads, mixing them with our antibodies, either from the rabbits or from mice or from humans in that case. Washing, adding the bioattenuated PD-1, adding streptavidin, phycloerythrin, washing. We did not do this because we found out that the system didn't work, but you could also then come in with the xMAP is a dual read system, and so you're able to come in with antibodies against our antibody, a secondary antibody against our antibody that's labeled with brilliant violet. And then that way you can read out the amount of antibody that's bound to the beads, directly or indirectly. And then the sample was ready to read on the machine. When we first did this, so this is our negative control. So this was using pertuzumab, which doesn't see the PD-1, of course, or PD-L1, of course. So we saw consistent high counts here. When we used a recombinant antibody to the PD-L1, we saw similar counts. And then when we tried our peptides, again, we saw similar counts. Very small amounts of inhibition. So for us, that meant that we were getting binding on the plates, but our antibodies were not binding well to the antigens that were on, once they were put onto the beads. So reasons we might try a reverse assay. So you talked about the reverse assay in the ELISA. Now this is reasons why we might try a reverse assay in this. So beads show binding to protein, but low binding of ligand. That wasn't the case in this. We definitely saw binding to the ligand, but Known blocking assays do not show binding to the beads. That was what we're seeing in that one. So we had a positive control that we know would bind to the beads or bind to the protein under normal circumstances, but that did not bind in there. And beads do not show good protein binding at all. And that can be because of the chemistry. I mean, any of you who have done any type of protein chemistry, especially binding things to columns and things like that, know that sometimes your chemistry doesn't work well with your protein. And the way it'll end up sticking to the beads or to whatever you're trying to stick it to will block interactions that you want to happen. So we design, so for my understanding, the method that we kind of worked on with some of the people at Luminex is something they've kind of done in-house, but nobody had published this yet. And so we developed this as a reverse blocking assay. So again, oops. We pre-mixed a biotin-labeled PD-L1 with our antibodies in the well. We had 25 microliters of each. So that would be sera, or diluted sera, and 50 microliters of beads. We do not wash. Very important. Because now we're going to add the beads. And the beads are added in. They were given an hour to incubate with the antibody-bound recombinant protein. Then we add the SAP in again. And now we can come in again with the brilliant violet labeled antibodies against our antibodies, wash and resuspend, and then the samples are ready to read. This was just showing, again, the reverse PD-L1 blocking assay that Linlin did, where we see rabbit IgG itself, very high counts, no blocking. The atezolizumab, low counts, which we did not see in that primary assay. So after a couple tries, we got everything working well. This readout is a percentage of inhibition of the biotin binding beads to the recombinant PD-1 coated beads. Here is our positive control. This is the atezolizumab. This was done as a percentage of the control. And what we see is our peptides at a much higher concentration of antibody do show blocking here. And these are the different peptides that Linlin had introduced in the previous things. And we're going to go through these very quickly. So this is the second readout that we did. We're showing here that we were seeing antibody binding to the PD-L1 on those samples that we went through. So this has to be where we had the primary beads bound to the PD-L1, which already had antibodies stuck to them. So this means that we do have binding of our antibodies to the protein, but it's incomplete in its blocking. And so that's why we're able to get a readout. And so you can use this type of a system to see whether or not your stuff is binding at all or not binding. With this, you can see the trastuzumab negative control, no binding. We had the positive control didn't work as well since it was such smaller levels. So I'd like to acknowledge Dr. Pravin Kalmaya. He did the concepts and the supervising on all this work. Linlin, who again worked with me on a lot of the animal experiments and did a lot of the in vitro work on his own. Heather Darby from Luminex helped us out when we were developing these original assays. Luminex Corporation, especially their scientific applications department and scientific marketing department, provides access to one of the IntelliFlex machines, as well as giving us reagents for doing a lot of these assays. Some slides were created with BioRender. We're supposed to say that. Again, all these assays that I'm showing here were produced on the xMAP IntelliFlex machine. And funding came from both the NIH and from Luminex. So Thank you.